We spent a bit of time looking at meristems when we looked at vegetative development, and we're back to meristems looking at the development of a flower. And the flower meristem is its own thing. So we'll see there's some genetic control where the plant signals to this developing part of its body that it's going to make flowers instead of leaves and more stem. And flowers are actually produced at the tips of meristems. So a flower is what we call a determinate structure, meaning it uses up the meristem as it's being made. So if you make a axillary bud that then culminates in a flower, then your axillary bud is going to develop these organs and nothing else. So another way to look at that is that nothing grows out of a flower. Flowers are basically an endpoint. They're produced to do fertilization, and then they develop into fruits if they have female parts. And that's kind of the end of what they do. They don't continue to grow any other organs from within. We can see at this level the order of the floral organs coming from the outside to the inside. Some of these are a little tricky to make sense of, but they're nicely labeled anyway. So here we have OT. This is outer tepals, kind of analogous to the sepals. And you can see there's 1, 2, 3. These organs are bigger. They were made earlier. Being made earlier means they're lower down and farther to the outside. So we're working from the outside in, also from the top to the bottom as we go through this. The next level in are the inner tepals. So kind of analogous to petals. 1, 2 buried in here, 3. So you can see the pattern. There's some symmetry here. Evenly distributed, 3 of these organ types. Moving in, ST, these are stamens, and we got 1, 2, 3, 4, 5, 6. So again, a nice symmetrical number. We're seeing organs in multiples of the same number, so 3 and 3 and 6. And these are developing, again, farther to the inside. And then finally, this kind of triangle here is developing into a set of fused carpels, it looks like. And that's going to continue fusing. So it's sort of an open feature at this point, but it's going to close up during development. But just at this stage, you can see everything kind of aligned in its order from the outside in. On the left side here, this is a flower of the grass family that's even more developed, or yeah, bigger definitely, with the same kind of organs identified here. So OT, outer tepals, IT, inner tepals, and then big stamens here, 1, 2, 3, 4, at least 4 in there, and then these fuzzy bits here. These are the stigmas that are going to collect the pollen. So pretty neat. I don't know. I enjoy looking at flowers as they develop. You can see the shapes and the numbers of organs established at this level that are going to be then visible to the naked eye as these things get bigger. So we'll take a few of these individually to look at them. The sepals and the petals really don't deserve much extra explanation here. They're pretty much modified leaves. So the sepals often are leaf-like. Petals are often colorful, but still leaf-like. But then the organs farther to the inside are a lot more highly modified for their functions in sexual reproduction. So we'll start with the stamens, which will be the organs that you would first encounter, the sexual organs moving from the outside to the inside. These are basically the male function, we call it. And their job is to produce and distribute pollen. So I mentioned before, they have a stalk known as the filament, and then toward the top is anthers. So anther, 2 filaments shown here. And we'll talk about how pollen is distributed in a different lecture, but you can see the pollen is produced and kind of sticks on here to the outside. So if you have a flower that's pollinated by an animal, the plant really wants to keep its pollen until an animal brushes against the pollen and takes it away. So it doesn't just drop it off into the into the world. Hold for pickup is the strategy for animal delivery of pollen. This image here in the upper left, this is a cross section through an anther, and this is kind of the typical structure for these. There's a lot of very similar features across almost all of the flowering plants. And this is one of those. So this portion here, this is the attachment point where this anther attaches to the filament. And then it's got these two chambers on either side, and that's pretty common. So the circular regions here, these are where the pollen grains are produced and they mature in there. And then as development happens, this whole anther is going to dry a little bit and tear open to release the pollen and make it available. Here's an example of that happening. The top images here, these are some just development of the pollen grains. So here, very early in the process, the PMC pollen mother cells, and then pollen grains a little more developed in this region. And then the colorful parts at the bottom, I like this illustration, really highlights how the anthers break open to release the pollen grains. So they have kind of a weak point programmed into them that as this dries out, tears open, and the covering kind of peels back for a lot of these, especially if it's animal pollinated. The process, this term up here, dehiscence, just refers to the process of splitting apart, splitting open. And a nice one here, this is a electron micrograph of the anther of a plant. This is the filament down here in the lower left. And some of that anther surface is still visible here. It's artificially colored. And then you just see lots and lots of these pollen grains. They're sticking to each other and really waiting to stick to an animal is what their function is going to be. Moving farther toward the inside, the carpels. We showed you one developing before. Now we've got a different species. This one has lots of different carpels that are all developing. They're still kind of open here, so this is another feature. This is one of the kind of a relative or maybe a good stand-in for one of the earliest flowering plants. Has lots of separate carpels, and you can see they're each kind of fusing themselves closed. That's one of the hallmarks of the angiosperms or the flowering plants. Angiosperm means the seeds are kept in a vessel. This is the vessel, the ovary. So it's going to close up there. You can see it's kind of finishing the closure. Closes up with a little seam almost here. And then this top fuzzy bit here, this is the stigma. This is going to be receptive to grab pollen grains after they're delivered by an animal. Within the carpels is an ovary. So here we've got a little bit different one. This is a Arabidopsis again, I believe on the left. And we're showing a big zone, which is the ovary. This is the chamber that has the ovules. And so the relative size of this top portion, this is where the pollen grains will be collected. It's the stigma, the sticky part where the pollen grains attach to. And then there's a little bit of tissue here that the pollen grains will, or the pollen tubes will grow through. And then the goal for the pollen is that it's going to get to these different ovules, so these kind of circular shapes in here. These are ovules, and they are waiting their turn for pollination. Different illustration here. This is an ovule, and you can see the connecting point there. So the ovules are connected to the tissue of the ovary. The pollen, we'll see it later, the pollen actually germinates and produces a tube of tissue that grows through the female tissue of the pistil or the carpel and grows toward each of these. Each pollen tube grows toward one of these ovules to do fertilization. So they're connected here. And then after fertilization, these ovules are going to become seeds, and they'll stay connected to receive some nutrients from the mother plant as they develop into seeds. And this whole thing around them is going to develop into a fruit. Okay, so developing a flower. We've seen some images of how it develops. Some other kind of putting it all together, part of this image. So we have a longitudinal section through the tip of a stem here. It's got that kind of dome that we saw before. We're familiar with the meristem, except this is now a flower meristem. So it's producing what kind of look like leaves at this level, but these are the sepals toward the outside, the petals toward the inside of that, stamens toward the inside of that. And they're all quite young at this stage, so nothing is quite finished developing. If you take it through a cross section, you can see the outer sepals, in from those, the petals, in from those, the stamens are blue, in from those, the carpels. And what this is going to transition into is we can talk about how these different kinds of organs are basically signaled by the plant as to where they're going to be. So this diagram at the right, number C here, is telling us about the kind of the genetic process, the developmental process where these fields are laid down. So if you have 4 kinds of whorls, sepals, petals, stamens, carpels, you're going to need 4 zones that are kind of established as a zone of identity. So field 1 is meant to show here like a combination of effects that stretches over the first 2 whorls. Field 2 is a combination of effects that stretches over whorls 2 and 3. And then field 3 is a combination of effects stretching across 3 and 4. This will make more sense as we get into the genes we're going to talk about with that. And here it is. So this is kind of the, this is a really neat achievement for a lot of scientists to have come up with this model and then validated it with a lot of genes. So the process of development, if you think about individual cells, we've got individual cells that are kind of waiting for their orders as development happens. And the cell says, well, what am I going to be, right? So the genes of development can say, you are going to become a petal, or part of the petal anyway, and you're going to become part of the stamen, and so on. And so those genes that establish the locations, for a long time, there has been this model called the ABC model, and I don't have A, B, and C on here, but we'll check it out in a next slide. So A, B, and C are these like types of genes that are kind of shown with these different blocks of color here. And they are, some of them overlapping, and the overlapping of signals gives us the signal for what kind of organ they're going to make. So let me take this zone here. It's got a variety of symbols, and maybe we'll just stick with the colors. And those then give you certain kinds of organs. So if you have a zone that's got just the AP1 green zone here, the zone that's only green, toward this outside, toward that side, all the way in a circle around the very outside, that goes down and that gets you sepals, so SE for sepals. The zone that has the blue overlapping with green also goes all the way around, and that causes petals to be made. The zone that's got blue and orange, that goes all the way around, and that produces stamens, ST. And this zone in the middle that's only orange, I'll kind of ignore the red for now. This only orange zone then gives us carpels, the center zone. So if you've got these zones of signaling basically laid out and they overlap a little bit, you can have these 3 colors or 3 types of signal to get you 4 types of organ. So blue alone, sepals, I'm sorry, green alone, sepals, green plus blue, petals, blue plus orange, stamens, orange only, carpels, 4 types. Okay, so I'm never really sure how much detail to go into on this. So the A, B, and C genes have been studied quite a bit, and it turns out they have probably a common ancestor. So there basically was an original set of signals, and it proliferated and became slightly different genes, and then they could specify different parts of the flower. So these are them with their abbreviations: AP1, AG, AP3, PI. I just highlighted a couple here that are in the A class, and the B class, and the C class. And this is an alignment of their proteins, and there is a lot of similarity. You just maybe have to take their word for it. But these zones that are highlighted, they have very similar amino acids making up the protein, kind of highlighting that they have a common ancestor and also common functions. So they're related proteins, but different enough that they can have different signals with different effects in the flower. So here's our evidence, diversification. And I've been talking about genetics. I'm trying to avoid going into too much genetic detail here, but These genes are what we call transcription factors. So them being present or sort of turned on in a region of flower development means that they can also turn on lots of other genes. So that if you need to make a stamen, it's not a matter of there's just a gene that does all the work to make a stamen. There's probably a whole set of genes that are important for making a stamen, and they can all be kind of turned on by this transcription factor that gets that done. So really cool stuff, but getting into the realm of genetics and a little farther away than some of you might be comfortable with. Same thing here. This is just kind of all the same concepts as the ABC model again. This one's highlighting the locations where these different genes are active in different developing parts of the flower. So I kind of like this part here. They're painted on. This is a inflorescence region. So this is a set of different developing flowers. FM is floral meristem. This is a developing flower. This is a developing flower. So we're looking at a set of several flowers that are all developing in here, and just highlighting the locations in those flowers where these different genes are being expressed. So B is part of what's going to make the stamens and also the petals. C is responsible for making the stamens and the carpels, so it's farthest toward the inside, a ring kind of in the middle for the B class, and then A farthest toward the outside. Again, just artificially painted on here, but this is a summary of lots of information that's already been known. Over here on the left, we've got longitudinal sections showing different regions. So sepals, petals, stamens, carpels, 4 different zones of the flower moving from the outside to the inside, and the specific genes and where they're active. So just yet another illustration of where these genetic signals are laying down the foundation for the structures they're going to code for. So some of the reason we know about all of these genes, or some of the first evidence, was to study mutated Arabidopsis plants. So again, this model organism that's received a lot of study. People have been studying mutants that cause different types of flowers. So they're not just weird and bizarre, but they're actually telling us something about what is genetically going wrong in these different ones. So on the top here, this is so-called wild type. This is a functional flower. It's got sepals to the outside, white petals, yellow stamens, and then the center pistil. And trying to show, I made this image a mirror image. You have to kind of imagine it filling around in a circle. So C towards the center, B outside of that, and A outside of that. All these kind of going in a full circle around. So A is the widest ring around the very outside, B is a ring around the middle, and then C is just a kind of smaller circle in the middle. And if A, B, and C are all present, we get sepal where only A is, we get petal where A and B overlap, we get stamen where B and C overlap, and we get carpel where only C is present. But what happens if one of A, B, or C is missing? So we'll go down here to the loss of C function. So C over here was responsible for carpels and also stamens. So you need C to produce carpels and stamens. In the absence of C, something interesting happens where A basically fills in. So the presence of C was kind of keeping A out of this area. C's gone, A fills in all the way across. And now we've got alternating zones of sepal where there's only A, and then petal where there's A and B. So it's sepal, petal, petal, sepal, sepal, petal, petal, sepal. And sure enough, that's what the flower looks like. So they have this mutant and they say, what the heck is going on with that? But if you understand the controls, then it's not just bizarre, but it's actually, you can kind of make sense of that and say, okay, well, it's sepals on the outside, then petals, then sepals again, then petals, then sepals on the very inside. I might have said petals too many times. But definitely no stamens and no carpels in this flower. Next option, we've lost the A function. So A over here was responsible for sepals and petals. Remember, it was kind of pushing back against C. So in the absence of A, C fills in all this space. So C takes over, and toward the very outside are carpels, that's C only, then stamens, B and C, then stamens again, B and C. This one's a little bit harder. I'll admit it's harder to kind of make sense of that one. But again, those who know what it's supposed to look like, they can kind of figure this one out. So carpels, stamens, and then carpels. And this one, that's basically only sexual organs and not the petals or sepals. Okay, last one is losing B function, maybe a little bit cleaner again now. B was interacting, so B plus A was petals and B plus C was stamens. In the absence of B, A and C keep to their corners, and it's just sepal, sepal, carpel, carpel. So sepals in the outside, no petals, no stamens, and then just carpel toward the inside. So it kind of ties all together. The control, the genetic control for making a flower. A little bit of an illustration for this is, so we saw some cases where, as in this example, we took the reproductive organs away and substituted in extra sepals and petals. So it's more leaf-like, more petal-like, without any reproductive parts. And turns out that has a little bit of application to plants you might be familiar with. A lot of plants you buy from a florist, if you get a flower for somebody for a holiday, a lot of times you get something like this, or garden plants are often bred to do this. These are mutants. So if you give somebody a rose like this, full of petals, is not really the natural state for a rose. On the right side here, this is a natural wild rose. Simple, 5 petals and a bunch of stamens. So if you're used to this one, you might be very surprised if someone got you a bouquet of roses that had a bunch of stamens in there. What's all this pollen doing in my rose? So these are related. These are both roses, and the origin of this mutated rose is in taking the stamens, which you can see are abundant, and kind of removing those and replacing them with petals. So extra petals, not so many stamens, gets you this presentation that a lot of people like, lots more petals, and maybe even a little cleaner presentation if people aren't fond of this grainy stamen appearance. So next time you get flowers for somebody, keep your eye out for mutants. So also genetic control. We talk a lot about the ABC genes that are responsible for producing flowers. There also are genes responsible for symmetry. So some flowers have radial symmetry where the organs are coming outwards from the center. It's sort of the same symmetry all around. One way to look at this radial symmetry is that you could take a pie slice out of this, and this petal would look about like this petal. But in contrast, you can have bilateral symmetry, as in this example, and this petal is very different from this petal. So we got 2 little petals at the top and 3 big petals at the bottom, and overall this flower has bilateral symmetry, meaning you couldn't take an equal pie slice out of this. You can only cut it in half to get a good mirror image left and right. And there's a gene responsible for this, or several genes. One of them that's been implicated is cycloidea. This is CYC here. So we have a normal version for this plant species, and then this would be a mutated version that kind of reverts back to a radial symmetry in the absence of that genetic control.